our third project webinar. We're going to go ahead and get started. It's 1 p.m. I wanted to let you know we will be recording this session for posting at a later date on our project website. And this is a webinar from the Sustainable Spotted Wing Drosophila Management Program. You can read more about our project at www.swdmanagement.org. My name is Hannah Burek. I'm the project director for the Sustainable SWD project, and I'm going to give you a brief overview of the mechanics of today's webinar, and then we'll transition into our content. We have a lot of information to share with you today. So the webinar will last approximately an hour, including questions. We shoot to be done at 2 p.m. We may go a little bit over to answer some additional questions if we have sufficient interest, but the content should last about an hour. We'll post a recording of this webinar at our project webpage by next week. Those of you who have registered for the webinar, so if you're attending here live, will receive an email notification about the posting. And I'll also email to folks who've emailed me directly the link once it's posted. I've gotten a number of questions about whether or not we would have pesticide credits for this session. We don't have pesticide credits, but if you live in a state where you can file requests after the fact, you can feel free to contact me at the email address listed here for documentation of your attendance. During the webinar, if you have a question, please type it in the chat box. You can type to the panelists or to the entire group, and I will ask them either at section breaks or at the end of the session. After the webinar, if you have questions, you can send them to my email, again, at hannahburek@NC State University. Today's presenters are myself, Hannah Burek, in the Department of Entomology and Plant Pathology at North Carolina State University, Ash Sayal in the Department of Entomology at University of Georgia, Dara Stockton at Cornell, based in Geneva, New York, and Anton Abrigo, who is at UC Davis, based in the Entomology and Nematology Department. As we move through the slides, the presenter who is speaking will be shown in a photograph in the upper right-hand corner of the slide, just so you know who is talking. I also want to acknowledge that a number of other colleagues have contributed content to this presentation, including Joanna Chu at UC Davis, Christelle Guido at University of Wisconsin, Rufus Isaacs at Michigan State, Catherine Swoboda Badarai at University of Nebraska, and Vaughn Walton at Oregon State University. The outline for today's webinar is as follows. I'll be giving a brief introduction, then we'll move on to talk about thermal limits, which will be led by Ash Sayal, Overwintering Biology and Ecology by Dara, Long-Range Movement and Population Biology by Antoine, and a discussion of local movement and distribution led by me. As I mentioned, this webinar is an output associated with a project that I am the director for entitled Sustainable Spotted Wing Drosophila Management for U.S. Fruit Crops. This is a project that was supported by USDA NIFA and includes a number of collaborators from across the United States as well as colleagues internationally. This is a 4-year project that we've extended for an additional year through the end of 2020, so we're continuing the work of our project with the goal of integrating spotted wing Drosophila management practices with the management practices that we already use for other pest species in the affected host crops, to reduce our reliance on insecticides as the only means of controlling spotted wing Drosophila, and then to deliver this information to you, our stakeholders, and to facilitate adoption of our recommendations. We've accomplished these goals through 3 interrelated objectives. The first, looking at implementing and evaluating management programs. The second, developing tactics and tools that predict spotted wing risk. And the third, optimizing management programs. What we're going to be discussing with you today are outputs associated with our objective 2, focused specifically on understanding the sources of spotted wing Drosophila populations between and during our growing seasons. And we further focused in on understanding the relationship of low and high temperatures with the flies, long distance movement and sources of spotted wing Drosophila invasions, and non-crop host use and local movement of spotted wing. Now I'm going to transition to Ash's section, the first section, where he'll be discussing thermal limits of spotted winged Drosophila. Ash?
Good afternoon. Today I'm going to talk about thermal limits of SWD and the temperatures low and high ranges that SWD can tolerate and how that tolerates. So SWD is a vinegar fly. It's native to Eastern Asia and Japan. It was first detected in the US in 2008 and since then has spread throughout the US in all major fruit producing regions of the US. This is basically a vinegar fly. It's very easy to identify as you look at the pictures here. The males have two dark spots on the wings. That's where the name spotted wing comes from. And the females have this very unique ovipositor that distinguishes this particular species within this entire group. As you can see in this picture, it's a very close relative to the Drosophila melanogaster. That melanogaster species does not have that ovipositor, whereas Suzuki has this very unique sclerotized ovipositor that these females utilize to puncture the skin of otherwise healthy fruit and deposit egg inside the fruit. As you can see here in the pictures, once egg is laid in the fruit, you can actually see those breathing tubes from the surface of the fruit, sometimes even with naked eye as well. Once egg is laid, the eggs hatch within 12 to 72 hours, and larvae feed inside the fruit, causing a lot of damage. Within a few days, the fruit becomes unmarketable. Overall damage of this fly within the US fruit systems has been estimated to be over $800 million per year. If you add the additional costs of management, it is close to $1 billion per year impact that this species has on our small fruit systems. Next. Originally, this fly, as I mentioned, is from Eastern Asia. That is where it evolved in these cool climates. And since then, it has spread across the world. As you can see, it has spread throughout the US, in some parts of South America, majority of Europe, and some other parts of Asia. Now the question is, since this fly regionally evolved in those mountainous regions of Eastern Asia, in those cooler climates, how does this fly survive and actually thrive in those wide range of climates that it has been found to cause damage in those fruit systems? These were those fundamental questions that we attempted to address in this project. Next, please. The basic questions that we addressed in this project were: what are the thermal tolerances of SWD? Do SWD flies survive over winter in all or some of the regions? If they survive, do they need to feed? What do they feed on if they do? If they survive, where are they located during the winter? Last question that we addressed: can we target those overwintering flies with control measures that we have at hand? Next, please. So now that this fly has spread in majority of North America, is from temperate to subtropical climates, ranging from Canada to Mexico. When you look at the temperature tolerances, it has a really wide temperature ranges. The comfort zone for this fly, or optimal range, is supposed to be between 68 to 86 degrees Fahrenheit. As temperature rise, it does affect overall biology and survival of this species. As temperatures increase above 30 degrees centigrade or 86 degrees Fahrenheit, it affects reproduction of this fly. And when the temperatures increase above 90 degrees, there's very limited survival of this fly. As you can see, we have divided our East Coast small food production region into 3 different sections. one a northern section, including New York, Michigan, and those parts of the country, then North Carolina, sort of middle area, and then southernmost areas, including Georgia and Florida. And temperatures between these regions vary significantly during the field season. The temperatures you see here, for example, in Athens, Georgia, and Gainesville, Florida, as they show 81 degrees Fahrenheit in July, these are mean temperatures. Actually, when you look at the temperature ranges during the day, we do experience above 100 degree Fahrenheit temperatures during the day on many days during the field season. Now, the question is what impact do those high temperatures have on this fly, and how does this fly survive in those high temperatures during the daytimes? Next, please. We did this lab experiment to look at 4 different temperatures where we had these 24 degrees, 27, 30, and 33 degrees. This was exposure of the adult flies, mated adult flies at these constant temperature exposure. And we found that as temperatures increase above 30 degrees, overall survivorship and reproduction, or egg production in this case, is significantly reduced. Next, please. So there was virtually no survivorship above 90 degrees Fahrenheit. And at lower temperatures, or overall percent development of the flies from larvae to pupae was significantly higher at 24 degrees. It decreased as temperatures increased from 24 to 30, and at 33 degree centigrades, there was no pupation at all. And same with the pupal development time also. Development time actually decreased as temperatures increased, and once it reached lethal temperatures, then obviously we did not see any development to adulthood. So after this experiment, we did a follow-up study. Now, in the first experiment, we exposed those mated flies to constant temperatures. We did a follow-up experiment where we exposed the larval developmental stage of this fly to see how high temperature exposure affects the development of this fly into adulthood. In this experiment, we selected that maximum 30 degrees centigrade, where we did observe some survivorship in the first experiment. As you can see here, we had overall egg viability was significantly decreased at 30 degrees centigrade as compared to the 24 degrees, which was control temperature. Here in this experiment, we did include another treatment where we did treat the adult females to a heat shock of 30 degrees centigrade for 4 hours before oviposition occurred, before they laid the eggs. And that heat shock treatment to those flies actually helped them and improved their egg viability significantly than the flies that were not exposed to the heat shock. This does show that these flies have the ability to acclimate to the high temperature conditions as well. At the second step of this experiment, the flies that survived in this 30 degree treatment, we paired them up in all possible combinations where we had benign males. Benign females means the males or females that were not exposed to heat or high temperature treatment. And then we had either male or female exposed to heat mated with the untreated male or female, and both males and females treated. And as you can see here, when either male or female or both were treated, obviously their progeny was significantly lower as compared to control. But the most interesting part here in the second figure is that when males were exposed to heat, their uh, overall progeny was significantly lower as compared to where only females were exposed to the heat. This does show that males are more vulnerable to heat exposure as compared to the females. Next, please. So the second part is how do these flies survive those extreme low temperatures? As I mentioned earlier, these flies can be found in wide range of temperatures, ranging from temperate to subtropical climates, anywhere from Canada to Mexico. However, there are lower thresholds as temperature decreases below 15 degrees centigrade. Their reproduction is significantly reduced. And when it decreases below 0 degrees centigrade, below freezing, their overall survivorship is extremely reduced to almost no survivorship at those temperatures. So when we look at the temperatures, again, within our US north-south different regions in Geneva, New York, say in January is about 23 degrees Fahrenheit, and Raleigh 40, Athens, Georgia 43, and Gainesville, Florida is 54. Again, in Geneva, these temperatures are mean. It can go even lower than that. So now we did some studies to see how do these flies survive those low temperatures. Next, please. And the questions that we ask, what biotic factors affect survival outcome at these extremely low temperatures of these flies that these flies are exposed to? Another question, how does cold tolerance develop in this species? How are they able to survive as we see populations every year in all those regions, even those where there are extreme cold temperatures during the winter? Next question, does the duration of acclimation affect survival outcome of these flies? The main factors that may allow these flies to survive during these extreme low temperatures may be, habitat, diet that they have available during those times, and overall biological parameters of these flies and the temperatures that they are exposed to. Next, please. So there were two studies done. There were lab-based studies to understand best forms of acclimation. One study, an experiment one, was done where flies were acclimated at a static temperature. Flies were acclimated at a single constant temperature, then subjected to extreme lows. And in the second experiment, flies were exposed to this gradually cooled extreme low temperatures. And then survival was assessed daily to see how this two different regimes of acclimation affect survival of these flies at extremely low temperatures. Next, please. So for the standard rearing, we use two morphs, summer morph and winter morphs. And the acclimation was done at 15 degrees centigrade. In general, as you can see in these, once they were subjected to 5 degrees centigrade, 0 degrees centigrade, and minus 5 degrees centigrade, the survival of these flies, both the summer morph and winter morph, it did increase when they were subjected to, or after they were subjected to acclimation at 15 degrees centigrade for 3 days. And this third line, where the summer morph was not exposed to acclimation at 15 degrees centigrade, they did not survive even at 5 degrees centigrade more than 3 days. Significantly, overall survival was significantly decreased after 3 days, whereas acclimated summer morph as well as winter morph did survive in significant proportions up to 35 days. However, at 0 degrees centigrade, even acclimated summer morph did not survive at all. Basically, even after 1 day, the survival was significantly reduced. However, winter marf did survive up to 14 days, and then they saw a sharp decline in survivorship at and below freezing, below 0 degrees centigrade. When we looked at -5 degrees centigrade, none of those populations survived even for 1 day. So that does show that any temperatures below 0 degrees are lethal to these flies. However, they do find some means to survive in those regions where temperatures actually go below freezing for during the winter time. So what strategies do these flies use to survive those temperatures? We did some additional studies. Next, please. So the second experiment that we did, which is kind of more close to what they experienced during the natural environment as temperatures change gradually, uh, and they do acclimate with those gradual temperatures. What we noticed is that when they were subjected to acclimation at gradually changing temperatures, their survivorship was significantly increased. Found significant decrease in mortality, much lower limits than previously observed. This suggests that significant overwintering capacity, particularly in protected environments, uh, especially in those leaf litter and other environments they may find in the field. Next, please. So another parameter that we looked at was activity of fly. So winter marf flies remain active up to -2.5 degrees centigrade, whereas summer marf flies enter into chill coma below 0 degrees centigrade. We did not see any activity below 0 degrees in those summer marf flies. This does indicate potential foraging during winter among acclimated flies. That's what keeps them going during the winter. Next, please. So to follow up on those lab-based studies, we did a field-based experiment in multiple states, including Maine, New York, Michigan, North Carolina, and Georgia, basically representing north to south all range temperatures that we've experienced within the United States. The goal was to determine the likelihood of SWD overwintering success in different regions. Detailed look at real temperature shifts and mortality effects in the controlled, well-replicated field study was done in this experiment. Next, please. So just to show you how we set this up, assay containers, we did develop this container shown in the figure here, which was 32-ounce plastic cup, which contained a soil core with some leaf litter, just to mimic the natural leaf litter type environment they might experience in the field. And we also provided an apple, a slice of apple that those flies could use as a food. Adults were released in that cup and covered with this fabric lid, and pupae were placed in that environment in those small paper bags, of small sachet bags. Next, please. And observations were made on survival of these flies weekly. And our results show that the survival significantly decreased over, or survival did decrease over 10 weeks period that observations were made. And as you can see here, survival after 20 days was significantly lower, had decreased. And this second figure B shows cumulative time below 0 degrees centigrade. As the below 0 degrees time increased to 20 days or higher, survival was significantly lower. Next, please. So in this experiment, we also assessed the effects of sex, life stage, and phenotype of those flies on survival and their ability to overwinter there. And our results show that females survived better than males during the winter, which have some physiological reasons, as females do have more fat body within the system for them to, which allows them to survive lower temperatures based on life stage. Adults survived better than the pupal stage. And also, when we looked at different phenotypes, summer morph versus uh, winter morph flies did survive better than the summer morph. Uh, Next. So just to take home message, high temperature is one of the limitations for these flies to survive. As temperatures increase above 90 degrees, they become lethal to those flies, and they cannot tolerate constant exposures to any temperatures above 90 degrees for a long time. They have to find some places where they find some cooler temperatures, such as thick wooded areas surrounding the uh, small fruit fields, or maybe within the thick canopy of the blueberry bushes or other fruit canopy, that they can find some lower temperatures than those constant exposure to high temperatures. Acclimation is important for overwintering survival of the flies, and that enables them to survive some of the apparently lethally low temperatures that we see in the regions where they occur during the winter. The speed of acclimation significantly affects the degree of cold hardiness that they display. If when they are they are acclimated at gradually changing temperatures or gradually decreasing temperatures, they do display better cold hardiness and are able to survive lower temperatures than when just sudden change in temperatures or acclimation at one constant temperature. Cold tolerant flies, they remain active at temperatures below freezing, as we have shown in our studies, and also feeding is required for long-term survival at cool temperatures. And it does show that they have access to something that they can feed on within the leaf litter or some other microclimates that they use to survive during the winter. With that, I will pass it on to the next speaker.
All right, we're going to transition to our second topic, overwintering biology and ecology. Dara is going to lead that section, and we'll pick up on that topic about what do they eat and what do they need to feed on to survive.
Hi guys, thanks for being here today. So I'm going to be picking up where Ash left off and kind of talk about how we're thinking about overwintering survival in general in these flies. So in addition to this general susceptibility at these different temperatures that Ash just finished describing, we've been interested in these additional abiotic factors within the landscape that could enable SWD to survive in like more extreme climates. So I'm up here in New York, about 30 minutes south of Lake Ontario. So we're pretty far north. Winter, I think, has already begun for us. So it's quite a challenge for us to identify where these insects are living in our region and other areas along the East Coast and in the Midwest and the Great Lakes region. This is a pretty significant question. So the data I'm going to be presenting in this section is going to focus on two things. One, access to food, and also protected microclimates, and how we think these two factors interact to drive survival in this area. Next slide. So this is our working model, and I just wanted to show it to you to walk you through how some of these things are interconnected. I won't spend too much time on it, but as you can see, it's pretty complicated, and there are a lot of factors we think are interacting to drive survival. And so when I talk about diet and habitat, I mean that habitat can act as a buffer between temperature and the insect, meaning that it can provide insulation and even dietary access, and that oftentimes the diet and the habitat are linked as resources. The diet itself can be an important factor in survival, particularly overwintering, because it can act as a cryoprotectant or even a resource for early season reproduction. So when we talk about overwintering, survival, we think that these two factors are really important to consider. Next slide. So focusing on diet, when we think about this fly, we have to think of it, we've originally primarily focused on it as a fresh fruit specialist, meaning that it specializes on ripening berries and other fruit that it has access to. So the problem with that, though, is that these things like raspberries and blueberries are scarce in the winter. They desiccate easily, they rot very easily, so they're not going to be available within the landscape, especially in northern regions after probably November, at which point other things are required to survive. And we know based on our laboratory assays that they have to survive probably. So we have to identify what resources they could possibly be using aside from fruit. Next slide. To do this, we started to think about Drosophila in general, and we know by looking at studies on even the model species Melanogaster that use of alternative resources in the landscape like fungi, flowers, sap flux, and just detritus or rotting plant material are really common among Drosophila. So we wanted to look at some of these alternatives for Suzuki'i, even as a fruit specialist, to determine whether they could use them and whether those resources are likely available to them in the fall and in the spring. Next slide. So I'm not going to show a lot of the data leading up to it, but we suspected that some of these resources shown here were important, things like fallen rotting fruit in the fall, and even things like resources like goose manure, chicken manure, which can be prevalent within the landscape, especially in our region where we have lots of lakes and rivers and areas where these birds tend to roost, and sometimes on our farms themselves. So we found data that these things are in fact used in the lab. Next slide. I wanted to show this because it shows some of the diversity of the resources that we've identified. We've gone and done collections on different resources available in the spring, including some of these wildflowers like white champion and Virginia waterleaf, dandelion, and even milkweed tissue that we found that the insects will lay eggs on and potentially reproduce in. We've also found that they will readily lay eggs in goose manure, and we found reproduction in that as well. And of course, wild fungi appear to be acceptable hosts for this species. So there's a lot of potential for these insects to use these different resources within the environment in addition to fruit, which might change how we think about looking for where we can detect them. Next slide. To complement this, there's been some research out of Michigan that we wanted to highlight. This was out of 2016, where they started focusing on fruit wastes and trying to identify these harborages of Suzuki'i on farms. And the long and short of it, if you look at this table, is that a number of fruit wastes are used, and sometimes very high numbers. So in particular, you can see compost and pumice are being readily used in addition to just fruit drop within the field. So these alternative resources are not something that we should be ignoring when we're trying to monitor for the pests during the off season and potentially control them. Next slide. That being said, though, we wanted to highlight that when it comes to development, that not all of these resources are equal. So although we can see that the insects will lay eggs, that does not necessarily mean that the larvae do very well. And in fact, in the lab, we found that although they will lay eggs in things like manure, that the adults, when they develop on them, there's very few in number, and the individuals are quite small in size, indicating that there was a good amount of stress involved in developing on those substrates. So even if they are going to accept these materials, the upside is that they don't do particularly well. So we need to determine how widespread this could be in nature and what that would mean for fitness consequences. And it could be that although they're acceptable resources, they're not widely used. Next slide. Some other research out of Oregon has shown what types of nutrient contents within diets are important to survival. And the long and short of these figures is that high carbohydrate diets are important for survival. So if you look at the yellow line in both of these figures, the winter morphs at the top and the summer morphs at the bottom, that high carbohydrate diets do the best, followed by increasing proportions of protein. So flies with a high protein diet do poorly. And this is consistent with our idea that this is a species that evolved on fruit, which is high in carbohydrate content. So that probably is maintained. So these alternative resources would still have to have a significant carbohydrate content, probably to sustain them through the winter. Next slide. Wanted to follow up by also just showing this slide, showing that although that's the case, we still have data showing that they probably do have to feed over the winter. So reiterating that diet's very important for looking for these flies in the winter. Next slide. The next step, and I just wanted to spend a couple minutes talking about this, is refuge use. We are probably a little further behind in terms of collecting data on this, but we're starting to really focus on it. We need to identify where to look for spotted wing. And so within the literature and throughout the past several years, we've been focused on a few different areas, understanding how things like snowpack and anthropogenic structures can act as insulators within the landscape, as well as more natural resources like pine cones and leaf litter, which can act like refuge for the flies. And then more recently, understanding how things like fruit waste can act as a heat source. Next slide. So in the lab, we've been investigating how these types of refuges may be used. So the very first figure I want to show on the upper right-hand corner is, it shows how the insects move into structures as temperature decrease. And there are two lines there, one's winter morphs and the other is summer morphs. And what we end up seeing is that for winter morph flies, they steadily enter structures as temperature decrease. And once we get to freezing, all of our insects end up hiding under these refuges. In contrast, unacclimated summer morph flies tend not to be able to do this. And that's because they tend to enter a chill coma before they've entered those structures, which further highlights the importance of working with populations that are in fact acclimated winter morph flies, because they seem uniquely suited to engage in these types of behaviors that we think are important for survival. But beyond understanding just what type of populations and what temperatures they would be moving into these structures, we performed some other experiments. So we think that they're probably forming these small aggregates in protected microclimates. So in the lab, we found about 50% of our insects or more tend to cluster in an individual spot. And that the places they tend to cluster share some common traits. They tend to be dark in color, black or red. And I didn't have space to show it here, but they tend to congregate low to the ground, regardless of food availability. So we think that, as has been suggested previously, areas low to the ground, like under leaf litter, are probably prime candidates for an overwintering location for these insects. Next slide. This slide, I wanted to point out that we are still struggling to detect where these insects are within landscape. So we've been trying to trap and detect them during the off season. And although we are able to detect Drosophila in general, and they tend to be in these forested areas, you can see in this figure, we have compost, coniferous forest, crop, and deciduous forest. Overwhelmingly, Drosophila are represented in forest wooded habitats. But detecting spotted wing, at least this far north, is proving really tricky. So we need to continue focusing on off-season habitat and refuge, and we're getting closer to this, but we need to continue doing that. Next slide. So some of our take-home messages are that overwintering is likely, even in extreme climates, but we think that this is in part due to mitigating factors of diet and habitat. Now we think that if we can identify the diets these flies are using in the wild, then we can potentially hone in on where they are and then target them. So going forward, it's important for us to be considering how this changing resource utilization by these flies occurs in the landscape, because it'll help us narrow in on our monitoring efforts and ultimately our control tactics. Next slide. The last thing I wanted to speak about was our continuing goals. And this ties into the first section that Ash presented on, but we really want to be at a point where we're able to predict infestation based on factors within the landscape and abiotic factors in preceding winter, meaning that things like, can we say when there's a mild winter preceding us, is the infestation risk going to be increased for the subsequent year? But simultaneously, if we know what landscape features are in the environment that are a high risk or say a low risk for SWD infestation, can we give growers a little more information about what their relative risks are and therefore give them better control tools? So I wanted to show this table that was modified from Heather Leach's more recent paper from Rufus Isaac's lab, and she was able to model and show that the peak infestation does vary based on the climatic conditions in the winter and spring preceding. So it's extremely important, I think, that we keep these things in mind, both as we continue on our research on overwintering biology and ecology, and as we're trying to develop these tools. But I think we're getting closer to honing in on the factors that are really important for overwintering survival, and we certainly have data at this point leading us to that. So that's all I have for today, Hannah.
All right, so now we're going to transition from talking about how temperature affects the ability of these flies to persist between seasons during the year and talk more about the movement of the flies. First, we're going to talk about long-range movement and how that and the implications that has for population biology, and then we'll transition into talking about local movement and what implications that has for fly distribution.
Hi everyone, thank you for attending this webinar. So first of all, I would like to just point out a few key goals that are really relevant to SWD management. So first of all, understanding SWD dispersal patterns between states could help us to predict, first of all, the spread of the insecticide resistance, if there is any. Second of all, the movement of genetically engineered SWD upon future release. And third, the spread of biological control agents upon future release as well. So second, some key goals of this project will also help us to understand the local adaptations and how that influence SWD populations and management, such as cold adaptations, insecticide resistance development, and other traits that we can think of and that could be linked to a diversity in the genomic background of the flies. And then like the genomic survey of the US geographical populations will also provide data on how the genetic variations necessary for the design of genetic pest control strategies and could inform on the risk of the insecticide resistance development and help us to develop new tools. Next, please. So basically, the experimental setup that we used was to collect or get flies that were sent to us from many different locations and extract the DNA from single flies in order to generate libraries for sequencing. Then that we submitted to a genome sequencing facility using the Illumina ISEC 4000 platform, and then using bioinformatic tools to help us identify SNPs, such as single nucleotide polymorphism, that can then be used to differentiate the different SWD populations and GWAS for traits. So then, based on those data, we were able to build some PC analysis to look at the relatedness of the SWD populations using, as the example here on the lower panel on the left, PCA, which is principal component analysis. Next, please. So far during this current project, so we were the first one to sequence the first Drosophila suzuki reference genome. And since then, we have sequenced samples from about 40 locations all over the world for then a total of 350 SWD genomes that will help us to gather data on the genetic diversity and the population dispersal. So far we have flies from 8 different states all across the US. So from Georgia, Oregon, New York, Michigan, North Carolina, California, Maine, and Washington. This data set will provide some information on the genetic diversity within the US and as well on the SWD dispersal strategy between the states. In addition to those 8 different states, we also have flies coming from 7 other countries all over the world. So from Europe, including Italy and Ireland, from Asia, in South Korea, China, but also from Brazil, from Hawaii, where the flies also originate in the US, and Japan. So those data sets will also provide some information then on invasion biology and being able to look at the invasive history of the flies. Next, please. So basically, this is just the map showing where all the different sites from the different United States the flies were collected from. So as you can see, mainly from the East Coast and the West Coast with different locations. And thanks again for sending us all those flies to include in this analysis. So next, please. The next will be like also a map of the different locations where the flies were collected all around the world. And then on the next slide, you will see a table that summarized different informations on the flies that were collected at those different locations with the GPS coordinates and the different date of collections for the different sites. So those, and as you can see, this code color will match the different flies, the different locations that we use for further analysis, such as the PCA, and look at the diversity of the flies. So you don't need to keep in mind all those colors, because you will see that the flies spread among different clusters. So on the next slide, this is the first PCA analysis that we perform to look at the population structure of Suzuki. So this analysis was performed on 1,007,000, more than 7,000 single nucleotide differences within the SWD samples. And as you can see on this graph, there is clearly 2 different clusters. One's restricted to the Western cluster and one to the Eastern cluster. So the cluster 1, the Western one include California, Washington, and Oregon, whereas the cluster 2, we have flies from Michigan, Maine, North Carolina, New York, and Virginia that cluster together. And based on this analysis, we can see as well that the flies collected from Brazil clusters pretty well with the US cluster 2, which is the Eastern one. What this analysis tells us as well is that SWD from all the other international locations are quite differentiated from each other, except for the cluster from Japan and South Korea that tend to cluster together. So to follow up on this analysis and confirm the result that we found using the PCA, we then perform TSNE, which is the T-distributed stochastic neighbor embedding analysis with the top of the 40 principal components. And basically what we can observe from this analysis is that the second method to analyze the structure of the different populations of SWD leads pretty much to the same conclusions, where we found again this first Eastern cluster and Western cluster, and then this Eastern cluster, where the Brazilian flies seems to cluster with. And again, we also found the Japan and the South Korean population clustering together, whereas all the other international locations are pretty well differentiated from each other. On the next slide, so we were also able to confirm those two approaches, those two results with another approaches using the DAPC, which is the discriminant analysis of principal components, where you can see here is the contributions of the different populations to each other among the cluster. So as you can see in the middle with this single color, which is green, yellow, and purple, that the Hawaiian, Ireland, and Italy populations are pretty well differentiated, whereas we found the same mix of color for the Eastern cluster and also the same mix of color for the Western populations. And as you can see on the very far left, the Brazilian populations that shows some characteristic of most of the US populations that make it think that yeah, they cluster pretty well with the US population. On the next slide, so then to follow up on this analysis and on this first batch of sequencing, we wanted to look at the seasonal analysis of the SWD populations. With these overall questions is SWD flies over winter in the northern locations, or are they migrating from warmer and southern locations every year? Which will be more relevant to the spread of the resistance, probably, of insecticide resistance. So basically, the experimental setup here is just comparing the genome of flies collecting from one year to another, from one season to another. And here you have the way how we collected the flies and how they were sent to us to look at differences from one season to another. So here we sequenced 10 individuals per time point for a total of 120 genome across all the locations that we look at and seasons. So right now, our bioinformatic analysis is still in progress, but we definitely expect some overwintering occurring in those populations. So just some take-home messages that I would like to give you. It's about the SWD populations within the US first. We found populations from California, Washington, and Oregon that are grouped together, forming the Eastern cluster, and that are pretty well differentiated from the Eastern cluster, composed by the Maine, Michigan, North Carolina, New York, and Virginia flies, and that are forming the Western cluster. Again, just some things to mention as well is that the Georgia populations share some characteristic with both the Eastern and the Western population. So we are not sure right now what's going on with these populations, but we will have like further informations once we gonna be done with the bioinformatic analysis for the seasonal sample. So again, the seasonal analysis for SWD from north to south location is still in progress, but we are kind of expecting some conclusions that will support overwintering in colder regions, and also that could lead to some bottleneck and local adaptations in those populations. So for the international populations of SWD, again, the Brazilian populations is for sure coming from Eastern US, and populations from Italy, Ireland, Japan, China, Korea, and Hawaii are pretty well differentiated, even though if we found the South Korea one and the Japanese one clustering together. So this is a pretty large data set since we already analyzed over like 350 genome, and just want you to keep in mind that this could provide unprecedented opportunities to look at local adaptations based on the various selective forces such as temperature, insecticide spray programs, host choice, and will also allow for the design of effective genetic control for SWD. Thank you.
All right, thank you. So now we're going to move on to the last section where we'll discuss local movement and distribution of flies within the landscape. And the key questions we have related to this section are: when during the year are flies active within an area? How does the surrounding landscape impact infestation within a crop? Where are flies found during the growing season? And where are flies found during the day? And I just first wanted to touch off by saying that we see a very consistent pattern of annual activity for spotted winged Drosophila. These are data just summarizing trap captures at North Carolina monitoring locations over the course of 6 years. And we see this consistent pattern where during the beginning of the year, we catch very low numbers, but we have flies present. And then by the time we get to the middle of the year, so right around the beginning of July on a consistent basis, is when we tend to see an exponential increase in fly populations. You can see that illustrated in the 211 trap captures, 212, 213 as we move, or 2013 as we move through. And so this is a very consistent pattern that we see. Where that exponential increase will occur will vary based on where in the country that you're located, but that's a relatively similar pattern regardless of where you're at in the United States. So first I'm going to walk you through some work that was done by Emma Pelton, who is a graduate student at the University of Wisconsin, and these slides are provided by Christelle Guido, who is her faculty mentor there. And what Emma was interested in was understanding whether the amount of woodland and the surrounding landscape affected the timing and population abundance of spotted wing. And so she she selected locations where there was a high percentage of woodland or a low percentage of woodland across 3 different states that varied widely. So she worked in multiple locations to replicate these conditions. And then she trapped flies throughout the growing season at these locations using the yeast and sugar baited traps shown in the upper right-hand corner. And what she found was that in locations where there were higher proportions of woodland, there were higher fly trap captures, but importantly, fly captures were higher outside the fields than they were in the crop. And this is a consistent pattern that you're going to see carrying through a number of different data sets that I'm going to share with you. So first take-home message is we catch more flies outside the crop than we do in the field in most cases. But woodland did have a significant effect on first detection of flies. And so when she controlled for state and year, and then the percentage of woodland, flies appeared 1 to 2 weeks earlier in areas that had higher proportions of wooded habitat surrounding them. However, this did not affect population abundance over the course of the entire season, meaning that flies became active earlier in locations that were surrounded by a lot of wooded areas, but they weren't necessarily supporting higher populations of the flies when everything was said and done. So Christelle's group went on to look at additional activity concerns. And so now I'm going to share with you some work done by Benjamin Yaffe and Christelle looking at daily activity patterns. And this is a similar experimental design to 2 different studies that I'm going to share with you after this, where they looked at time periods during the day and trapped flies, either using baited traps here on the left or passive traps, these clear sticky traps. So the baited traps had an attractive lure and the passive sticky traps were just a sticky matrix. And they looked at these trap captures over different chunks of time throughout the day to figure out when the flies were most active. And at their locations, Ben and Christelle found that spotted winged Drosophila trap capture was highest in the evening, so 6 to 8 p.m. And looking at those passive traps, they replaced these over those same 2 to 3 hour periods, except overnight. They found an interaction between trap height, so whether they were in the middle of the canopy, the ground, or the top of the canopy, and time of day. So they found more trap captures in the early hours and the evening hours, and they found higher trap captures at the middle of the plant as opposed to either at the ground or the top of the canopy. So to further parse this interaction, they looked at where those flies were going at those different times of day. And what they saw was that in the morning hours, 9 a.m. to 12 p.m., there were more flies in the middle part of the canopy. And then the top of the canopy trap captures increased later in the day. So there was a flip from low trap captures early in the day to the top of the canopy in the evening. So there was variability as to where the flies were based on the time of day as well as the space within the plant. So interestingly, they also extended this out to look at where some non-target insects might be as well. So they looked at pollinator activity in those same areas of the plant, and they found that pollinators were concentrated at the top of the canopy regardless of the time of day. And so this has some implications when we might be timing management activities for our spotted wing and trying to minimize effects on non-target insects. I'm now going to show you work from 2 other cropping systems looking at a similar question: when during the day are these flies active? So this is work done by Ash's group down at University of Georgia in blueberries. And again, they had these similar blocks of the day, either dawn, day, dusk, or night, in which they used, in their case, these traps baited with yeast and sugar attractants across a grid in blueberry traps, either outside the field, on the border of the field, or inside the field. And what they saw was similar, that trap captures were higher in the field at dawn and at dusk, and they differed across those locations. So the same pattern was found at the border locations, and in the wood locations, there was little difference. And so they saw a significant difference between the locations where the traps were found or placed and an interaction with the time of day. And so that dawn dusk activity was more strongly associated with crop locations rather than edge locations. So finally, I'm going to show you work that was done in my laboratory by Catherine Swoboda-Badarai, who looked at blackberry populations. And so she had traps that were on the edge of the field in blue or the interior of the field in red. And these were the yeast and sugar traps. And what she found was that, again, trap captures were highest for her on the edge of the fields, and trap captures were greatest at dawn and dusk. So this is hour of the day on the x-axis, and total trap captures on the y-axis. And during periods of dark, there were essentially no trap captures, and the highest trap captures occurred right before dark and right after dawn. And similarly, we found more flies on the field edges than we did inside the field. So Katie carried this out to a 3rd step where she looked at what implications that had for fruit infestation. And she caged fruit and exposed these, so she caged fruit prior to infestation occurring, left those cages on while the fruit were allowed to ripen, and then exposed those fruit during those same observation periods, and then placed the cage back on. Subsequently collected the fruit and looked at the number of eggs that were being laid. And so what she found was that egg laying patterns lined up with those fly activity patterns. So we had higher proportions of berries infested at dusk and dawn than we did during the middle of the day, and none occurring during dark. And then the rates of infestation for berry were higher at dusk and dawn than they were at other times of the day. So the fly trap captures are mirroring fruit infestation patterns. Katie was also interested in understanding whether proximity to non-crop habitat affected fruit infestation levels. And the strategy she went about was to look at fruit infestation in samples of fruit, approximately 40 ripe fruit, gridded across field locations. She worked at a total of 4 different blackberry locations across 2 years. At each of these points on the map, she had a monitoring trap for flies, as well as those 40 berry samples. And I really like this set of slides just to illustrate how an infestation develops over the course of a field scale over time. And so Katie went out as soon as the fruit started to change color and began collecting these 40 berry samples and rating the degree of infestation. On her first observation date, when ripe fruit were first available, there was no infestation observed. The following week, a few of the locations across the field, randomly distributed, had 1 to 2 pupae per 40 berry sample. The following week, every single location had every single berry infested with greater than 1 larva per berry. The 4th week of sampling, fruit harvest had tapered off and infestation was also waning, with fewer pupae than the third sample date and a couple locations no longer having infestation present. And this seemed to be tied more to the decrease in fruit availability than anything else. And so this just illustrates the rapidity with which an infestation can develop, and that infestation is not necessarily linked to fly trap captures across space. And the reason I say that is because this is the trap capture data for a single transect from this field. So this is spot 201 through spot 209, and these are the traps at 201 through 209. Trap 201 was located outside the field, and again we see this consistent pattern where our trap captures are generally higher outside crop fields, and as soon as we get inside the crop where the growers were actively managing their planting, our trap captures decrease. And so as we move inward, our trap captures go down, despite the fact that we had 100% infestation along those same observation points. So the take-home messages from this section: surrounding habitat may have an impact on how early fly activity occurs, but it does not necessarily have an impact on population size or on fruit infestation patterns. Adult flies are found in higher numbers on the edges of fields, and they're active at dawn and dusk, and egg laying appears to occur at these same times of day. Flies can be found in different parts of the plant during different times of day, and infestation builds rapidly in fruit when susceptible fruit are present, and is not necessarily related to either location within the field or fly trap captures within that location. We think flies are moving in and out of fields to utilize those fruit resources for egg laying, but perhaps spending more of their time elsewhere. That concludes the research presentation component of our webinar. I did want to highlight some of our existing online resources, particularly our project webpage, swdmanagement.org, where our webinar recording will be posted by next week. If you are registered and attending this session, you'll receive a link to that recording when it is live. I also want to highlight some of our social media platforms as well as blogs associated with our project. If you have any questions or feedback, you can type them in the chat box. We'll hang around for the next 5 minutes or so and try to answer questions. Or you can also send thoughts on our project, this webinar, or other activities to me via email, and I'll share them with the team. Thank you all so much for attending, and if you have any questions, we will take them now. So we have a question in the chat box as to whether or not are overwintering populations the reason we are seeing more SWD earlier. Dara, do you want to take that one?
Sure, Hannah, let me see. Let me read this. Is this from Robert Alex?
Oh, sorry. Let me click over there. So from Jim O'Connell, there's a question in the chat box about overwintering populations. Are they the reason why we're seeing more spotted wing earlier?
Well, that's a good question. Based on all of the data we've been collecting from our group up in New York, with groups in Michigan, North Carolina, everyone that's been working on this, we're pointing in the direction of, we think there probably are small pockets of overwintering flies. Where they are, we're not sure. Unfortunately, that's been really difficult for us to detect. So I would say that earlier infestation could be related to overwintering populations, but we still need a lot more information. I don't, Greg, do you have any thoughts on this?
I mean, I would just add, I guess, that in years where we expect better chances of survivorship, we do see this correlation with earlier and higher infestation levels. And that's true in the Michigan State, I would guess. Hannah, if she looked at her data this way, may see the same thing. And certainly in Europe, which is, got that cold temperate climate, they see the same thing. Mild winters, earlier infestations, harsh winters, and we have to discuss how to define what we mean by harsh winters, but generally really cold temperatures over extended time periods. They have later arrival and lower overall density. So I think there's a connection. If we could figure that out, it would be nice to be able to provide some early warning of risk for certain regions. And I think that's one of the goals that we see going down the road.
Yeah, and I would echo those comments based on the early work that is coming out of Michigan State through Heather Leach and now Rufus Isaacs carrying on, that winter temperatures certainly have an impact on how early we would see flies and to an extent maybe how large the populations develop. For us here in North Carolina, a cool winter temperature is going to have an effect on reducing our early season populations and maybe delaying that population explosion that we consistently see. And a warm winter may result in earlier activity, which would impact some of our early harvested crops, strawberries and blueberries. But for our later harvested crops, for us, those would be blackberries and raspberries. Our population explosion is going to kind of overwhelm the effects of the winter. So for where we're located, winter temperatures are going to have the biggest impact on early season crops. But by the time our late season crops fruiting, any effect of the winter is probably washed out at that point and overwhelmed by reproduction. So I did want to address one very quick question as long as I'm talking, which was in the last study, was the blackberry field treated with any insecticide for spotted wing management? And the answer is yes. That was a commercial field that was being commercially managed. That was a challenging production year because of rainfall and other factors, but that field was getting weekly insecticide applications and still experienced a 100% infestation event. I'm gonna pull up. So the next question I have in the chat box is from Frank Hale, which is, do you see spotted wing infestations in strawberries in the mid to deep south since they fruit before July? And the answer to that, Frank, is that it is pretty variable and it's related more to winter temperatures than in our later fruiting crops. So strawberries in the southeast are harvested in early June, some in May, some in April. And in those times of year, we only tend to see spotted wing infestation at economically significant levels if we've had a very mild winter. All right, so the next question I see is there any information on the effects of a late spring frost on populations? Does anyone want to jump in and handle that?
Hannah, I don't think that we have any information on that specifically. I'm not sure. I don't think nothing's coming to mind for us.
Maybe in Oregon, they may have seen some of this stuff, but I don't have a good I'm not unaware of data that directly looks at that, but we do know that early season climate can affect the rates of population growth, and the modeling suggests that, and just sort of anecdotal observations kind of support that idea that the conditions are going to affect potential for population growth.
All right, so I have one more question we're going to answer, and then we're going to say goodbye for the afternoon. I am going to go ahead and copy and paste the questions in the chat box, and I will try to follow up with some of you individually. The last question I'm hoping Ash can tackle, which is, what is the duration of hot temperatures needed for spotted wing Drosophila mortality?
The duration of exposure for mortality, it actually depends, especially when it comes to field conditions. It's very hard to determine, as there are lots of places within the field that have variable temperatures or lower temperatures than you see out in the field. But we have observed, based on our trapping, that flies, when it's really hot outside, they do find some cooler spots either within the field or most likely out in the wooded areas, that they kind of slow down and sit there and wait until more optimum temperatures do exist. However, when the temperatures are above 90 degrees and flies are exposed to constant temperatures, they can die really within 24 hours. The majority of the flies would be dead.
All right, well, in the interest of time, I'm going to go ahead and conclude our question and answer session, but I do have the questions that have been typed, and I'll stick around for a little bit and try to answer them one-on-one, and keep a record of that to reach out to folks individually. Thank you again for attending, and the recording will be posted within the week.